Good afternoon, and my name is Austin Wei. Welcome to my presentation. I will be discussing my research project that I conducted at the Institute for Bioscience and Biotechnology Research over the summer, which is titled Investigating the Effect of Mutation in Cases of Parallel Adaptation. This is a basic overview of the content that I'll be covering today. First, we have to go through background, the hypothesis developed from the background, my methods and materials for the project, my results that I achieved, and my conclusion of the results, and finally acknowledgements. And after the acknowledgements, I will be answering any questions that you have for me. So currently, there are two arguments for how evolution developed. These two views may be likened to a comparison of a buffet versus a sushi conveyor belt. In a buffet, all possible choices are present, which means any mutation has the same probability of being chosen. What chooses the mutation completely depends on you and only you. So if you want, say, in a buffet, you go to a buffet, you want, say, chicken, you pick chicken. You don't pick beef, you pick chicken. Same thing, you want like a vegetable, you pick broccoli over cauliflower. In here, the concept of a mutation rate, or the concept that a mutation is picked more than another, is irrelevant. Alright, for the second one, it's called the sushi conveyor belt. Imagine like some sort of like a sushi belt that like almost never ends. And then so mutations or choices are being moved across your window, and then you only have a small time to pick your choice. And the rate of incidence and the probability of appearing will preference. So it means if you want beef, except you only see chicken moving across the middle, you might pick chicken because you want to have a meat, except you have no idea when a beef's coming, so you might have to pick chicken, even though there might be a beef along the way. That's a picture of the law of probability, where, we'll disregard that. Alright, so the big difference between these two models is how they view the role of mutation in the rate of evolution. In the buffet view, is each mutation has the same chance of being picked. In the conveyor belt view, different mutations have different chances of being picked, as different mutations appear more than others, and as a result, mutations that appear more often are more likely to be chosen because of a bias. And so to investigate this, I decided to study mutation bias. And the one mutation bias that I studied that can be easily identified through nucleotide sequences is called the transition transversion bias. Right, so transition mutations are mutations from a purine to another purines, or such as an A to G or a C to T. A transversion mutation is mutation from an A to C or G to T. From a purine to pyrimidine, or vice versa, pyrimidine to purine. Alright, so here you can see that there are 4 possible transversion mutations, and there are 2 possible transition mutations. As a result, you might think that transversion mutations occur more often than transition mutations. False. Theoretically, as I said before, transition mutations are twice as likely to occur as transition mutations. However, transition mutations are known to occur by a several-fold bias more often than transition mutations. I don't know why, but as a result, transition mutations appear more often in nature than transversion mutations do. So, the results of the study can possibly provide more information on how evolution, or specifically parallel adaptation, which were the subject of cases I studied, works. And so from my background, I developed two hypotheses, one for the buffet model and one for the sushi model. The buffet model became my null hypothesis. My null hypothesis is as follows. If the mutation rates affect the overall rate of evolution, then the transversion mutation ratio should be 1 to 2. 1 for transition, 2 for transversion, given that no bias is present, as there are twice as many transversion mutations than transition mutations, logically becoming a 2 to 4 or as 1 to 2. However, if mutation rates are factored in, my alternate hypothesis is if mutational rates affect the overall rate of evolution, then the transversion mutation is typically greater than 1 to 2, which basically means that the transversion mutation is greater than what the theoretical model is, which is 2 transition mutations, 4 transversion mutations for every nucleotide mutation. It becomes more than 1 to 2 because transition mutations are known to occur more often than transversion mutations, despite being only 2 possible for transition and 4 possible for transversion. Therein lies the bias, where I'm investigating. Alright, so for the methods, my methods are twofold. So, an authoritative review called Paralytic Genotypic Adaptation, when evolution repeats itself, authored by Wood in 2005, was examined. We decided to narrow the cases down to one single table, which I'll be showing later. The cases presented were then picked through 2 requirements: whether the paper was accessible as in like whether or not I could actually read it because I needed to see the data, and was the data presented to the table usable and accessible, such as if it was accessible online in PubMed, because I needed to see the actual mutations. And as a result of this filtering, I ended up with 8 studies. Alright, once I found those 8 studies, I acquired the data through the references provided by each of the authors in each of the papers, usually through PubMed Online. It's a site where you can find various nucleotide sequences cited in various papers. And then once I found the data on PubMed, I compared the data to the data inside the paper to see if those are the same data, because oftentimes there are multiple variations of a single nucleotide sequence. I needed to find out if the one I had was the correct one. Alright, so after that, mutations in each of the data were determined if they were transition or transversion mutations, and then tallied up and made into a ratio. And the statistical following in the form of a chi-square test would follow, comparing the transversion mutation ratio to the theoretical ratio of 1 to 2. This is the table I was referring to. As you can see, there are multiple different cases here. The table organizes itself in the form of a taxonomic group, which is the organism that they were studying. The phenotype, which was the trait that the mutation occurred in. Type of evidence, what mutation happens, such as amino acid substitution, nucleotide substitution, and then the reference was what paper it was. So, for example, the taxonomic group here is Mason-Cockelberg, which trait exhibited was herbicide resistance. The mutation was amino acid substitution, and the paper was written by Bernasconi. So that means Bernasconi studied the herbicide resistance that occurred through an amino acid substitution in Mason-Cockelberg. Alright, this is an example of the data I occurred. So as you can see, the black boxes surrounded this that are surrounding the letters represent a mutation. As you can see, I found 4 of them in a row of, say, 500 sequences. And then usually the sequences are, this is only a fraction of the sequence of one paper. Usually there are at least 50,000 nucleotide changes long. So I had to go through each and every one to find which mutations and what type of mutations they were. The results. My end tally resulted in a total number of 66 mutations over all eight papers, and then the number of transition mutations I found were 41, compared to the number of transvergent mutations was at 25. This results in a transvergent mutation ratio of 1.61 to 1, which shows that in this case, transition mutations occurred more often than transvergent mutations. This is a data table of my. As you can see, I found 4 transition mutations and 3 transversion mutations in the Bernasconi paper, which totaled up to 7. And then I added it all up to get 66. My results. This is the results of my chi-square testing. The TI and the TV EXP result represents the expected ratio of 1 to 2 out of the 7. So if I expected a 1 to 2 ratio of 7 mutations, and in 7 mutations, I would have expected 2.33 transition mutations and 4.67 transversion mutations. But these are the actual values I found, which resulted. So let's say Bernasconi, the resulting chi-square value was a 1.79, and then so chi-square testing resulted in an overall value of 24.623 with 7 degrees of freedom. Given that the p-value for a chi-square of 24.63 is 0.01, which is less than the 0.05 area of rejection, then the null hypothesis is to be rejected. This, however, does not signify that the alternate hypothesis is accepted. However, overall, these results tend to support my alternate hypothesis, in which it says that mutation rates are factored into the rate of evolution, and the mutation transvergent mutation usually is greater than 1 to 2. So we conclude that we can reject the model in which mutation rates are said to not affect the evolution rates. And data suggests that the effective mutation mass is more consistent with the second model the alternate hypothesis. However, in order to conclusively conclude that mutation bias does affect parallel adaptation, more experiments have to be conducted, or more investigations to be conducted, as I had a very, very small sample size. It's of only 8 cases. Ideally, you should have probably more than 100, and then probably compile data over time to see that if it's consistent, and different types of organisms have to be factored in, because different types of organisms have different types of mutations, and then multiple different researchers have to be considered, because researchers try to be objective, but eventually there's always going to be some small bias in where the researcher is looking for an answer and only finds that answer. So you have to have multiple different cases over multiple different organisms, over multiple different mutations, over multiple different researches to get a very, very conclusive and entire result. Acknowledgements. I would like to thank Dr. Arlen Stoltzfus of IBBR, Institute for Bioscience and Biotechnology Research, for allowing me to work under him over this summer. And then I would like to thank Poolesville High School for providing me with this opportunity to study and research in an actual lab environment over the summer. Do you have any questions?